We are here in person and online in downtown Vancouver, which is on the traditional, ancestral, and unceded territory of the Coast Salish people, the Musqueam, Squamish, and Tsleil-Waututh nations, who have been custodians of this land for thousands of years. And I am absolutely thrilled to open our 2025 Genomics Forum, where we will explore the real-world impacts of genomics on precision health. And this year's theme, From Research to Results, Recognizing Genomic Success in BC, will guide us through the incredible work that is shaping the future of healthcare here in this province and beyond. And at Genome BC, we're proud that the work our community has done in transforming healthcare through genomics, as we see it driving advances in diagnosis, treatment, and overall patient outcomes. And this year's forum is particularly important. It is our 25th year of existence and working in this province and making investments in world-class research and translating that into real-world impacts. And we've come a long way. We were talking earlier with Marco, who was part of the Human Genome Mapping Project that was published in 2000, 2001. Come a long way, so much more to do. And so that this milestone, 25 years of Genome BC being here, means that we need to look to the future. And our focus remains on what's happening now and what's next in genomics, and how these innovations will continue to shape the future of healthcare. So today we'll explore critical areas where genomics is already making an impact. You've seen our program. We're going to look at precision medicine, advancing cancer care, pharmacogenomics, and organ transplantation, rare diseases and diagnostic innovation, accelerating early and accurate diagnosis, maternal and child health genomics, improving care for expectant mothers and newborns. And breakthroughs in these areas are leading to improved health outcomes. They lead us to a future where healthcare is tailored to the unique genetic makeup of each person and patient. BC's strong research institutions and a thriving biotech industry have positioned BC as a leader in this space, and Genome BC is proud to play a role as a funder and facilitator of innovative research, all which require new partnerships. And because none of this would be possible without the collaboration of our scientists, healthcare professionals, policymakers, and industry leaders. So throughout the day, we'll explore the opportunities and challenges that come with the integration of genomics into healthcare and the broader world. And this forum is an opportunity for all of us to reflect on how we can continue to make a difference through innovation, collaboration, and responsible development. So I'm looking forward to the discussions ahead and to hearing from our speakers and panelists as they share their expertise and experience and a vision for what is possible in this province around precision medicine. So it's now my pleasure to introduce Genome BC Chief Scientific Officer and Vice President of Research and Innovation, Dr. Federica De Palma, who will introduce our next speaker.
Welcome, everyone. Thank you, Suzanne, and thank you, Shelley. That was wonderful. So welcome, everyone, for this fabulous day. I just wanted to let you know that it's quite special today to have all these leaders from our ecosystem together in this room, and some of them online. And the reason is I have started at Genome BC in 2020, and this is the first time that this genomic forum, at least since I have started, is in person. So I think it's quite special that we are able to have this hybrid format and still have a few people gather in person for this event. So I have to make a few announcements. So we have many speakers, and we are going to have a number of panel discussions, which, as you heard, are very insightful. But we don't want you to just listen to them. And we want this to be a two-way conversation. So there's going to be this wonderful room. All of you have microphones. So if you need to ask questions, and we do want you to ask questions, please use the microphone. And if you are online, please use Slido. Each session will have a Q&A, so it will have opportunity to include your voice. We also have we also have a series of poll questions, interactive poll questions. And so we will be asking your input through these. They will be introduced at the start of each session, so to give you an opportunity to respond and to vote. And then the panelists will be able to see the result and sort of react in real time to these. We will use a Slido once again, so make sure that you do log in with the details. And before I move on to the first poll question, which will open up this first session, I just want to remind everybody that the internet can be accessed as a login information. There are at the side of each of these, AG showing them, and is SFU net. You need to select from the available networks. Okay, so I think we are ready for the first poll question, which will be for our keynote address. And so we are asking, this is the question. In your view, what is the most important factor for successfully integrating genomics into the healthcare systems? Please follow the directions on your screens. If you're here in the room or you're on your screen at home, log into Slido and provide a response. I'll just pause for a minute or less than a minute to help you do this, give you some time. And I find this poll very useful and a nice way to interact throughout the session. And I also think they are wonderful for the people online as well who cannot be here today with us. Okay, so hopefully you're well on the way to answer, but I can't not think of a better person to open this session today if we are talking about leadership in genomics and, of course, how through genomics, the transformation of our healthcare system and precision medicine. And that person, of course, is Dr. Marra. Marco is a UBC professor of medical genetics and the Michael Smith Laboratories, is formerly the head of medical genetics and co-founder and past director of the Canada Michael Smith Genome Sciences Center for more than 2 decades. Dr. Marra is known for his work in genomics and precision cancer genomic medicine. And now I have a little anecdote that I'd like to share with you. Because Marco doesn't know this, there must be many people like me out there. But Marco was at the forefront of the Human Genome Project. And particularly what he did is he published these papers on developing this map-based fingerprinting approach earlier on, which transformed sort of the way we navigated and mapped the genome. Well, at the time, a few years after these papers were published, I'd finished my PhD, I started my postdoc, I was at the NIH, and then I went beyond that to Massachusetts. And every single supervisor of mine handed me his papers and then asked this is the gold standard now, this is what we need to do, not just for human, not just for mouse, for any species, any model species, please make it work and scale it up. And so I just want to say that for a significant part of my early career, Marco's name was associated with a fair amount of pain and suffering. It was respectful suffering, but nevertheless, it was a suffering. So I just thought I'll share this with you. Anyway, Dr. Marra was recruited to Vancouver shortly after he did all of this by Nobel laureate Dr. Michael Smith, where he led the teams that contributed to the sequencing of model organism genomes. From Vancouver, Marco sought to drive the expansion of genomics in Canada. He led and co-led numerous cancer projects. He revealed new mutations, biomarkers, therapeutic targets in many cancers, ovarian cancers, B-cell lymphomas, brain cancer, and the list goes on, and is very long. In 2003, he led efforts that generated the first sequence of the agent causing SARS, revealing it to be a coronavirus, and demonstrated that genome sequencing of a pathogen could be a key part of a rapid response to an emerging infectious disease. And that was key to respond to the latest pandemic. Dr. Marra currently co-leads the first program to demonstrate the use of whole genome and transcriptome sequencing to personalize cancer medicine. The program includes 80% of medical oncologists in BC and recruited thousands of patients with advanced cancers and has yielded over 50 manuscripts describing how whole genome and transcriptome sequencing can actually influence clinical treatment decisions, therefore seeing real benefits to cancer patients and their family in actions. Marco's recognitions are numerous. I have a few. They are not, and it's not a complete list, But Marco is a National Killian Prize in Health Science professor, UBC University Killian professor, officer of the Order of Canada, Canadian Medical Hall of Fame, Don Ricks Lifetime Achievement Award, awardee, the Outstanding Achievement in Cancer Research Award, the Two-Way Memorial Prize in Cancer Research, the Terry Fox Medal, the Order of British Columbia, the Merck Frost Prize. I'll stop here. I think we are very privileged to hear from Marco this morning.
Thank you very much, Federica. I had not appreciated that my papers were associated with significant pain. I'm going to have to be careful here and appreciate the anxiety that I've caused. It's a huge pleasure to be here today. Thank you very much for the invitation to speak. I'm going to start with a disclosure slide. I'm going to spend some time talking about the Terry Fox Research Institute's Marathon of Hope Cancer Centers network. And so best to get this all out front and center. I'm a deep believer in the vision and have various tasks and privileges associated with being part of the Marathon of Hope Cancer Centers network. So I think this talk was meant to be a bit of a sweeping overview of how things came to be as they are today. And I'm not going to do that, at least not very well. I started to think about how I could approach it all. And I was looking back on the past 25 years of what's happened in the province. And I came to the conclusion that I couldn't do the topic justice. And so rather than spend the bulk of my remarks on things that I don't think I could address well, I thought I'd do a brief overview, lay some groundwork, and then dig straight into some data and introduce the network. And the reason I want to introduce the network is because I think it's a useful framework to think about how one can scale genomics, at least in one context, nationally. And we can talk about the challenges that we've experienced in the various jurisdictions that make up the country, and we haven't confronted them all successfully yet. So there are more challenges in front of us. I think the first Slido question was very provocative to me. I was paying attention to that, and I thought, you know, the answer that I would prefer there wasn't, in fact. And so I would say that if you want to get genomics into healthcare, you need to have genomics leaders that see the potential of healthcare and want to support healthcare. And you need healthcare leaders that see the potential of genomics, want to nurture genomics and grow genomics into the healthcare space. That's what you need. And if you have the leadership right at the beginning, right at the start, and leadership can come together to make this thing happen, the pieces will eventually fall into line. And it won't necessarily be quickly or easily, but it's motivation that starts at the top. So that would be the answer, or my answer to the first poll question. Okay, I tried to think of a way to encapsulate the history of genomics from my perspective anyways, in a single slide, and this is the best that I could come up with. So I've decided that I could defend the concept of genomics, our history, broken down into 3 eras. These are not mutually exclusive timeframes or anything of that sort, but maybe this is a useful way to start our conversation together. So Federica mentioned this. This is the source of much of Federica's pain, I have now come to understand from those brief remarks. And that was the reference genome era. And so the idea was that in the reference genome era, we could sequence the genome, a genome, and we would call it the genome. And so this concept started with model organism genomes, Caenorhabditis elegans, Drosophila yeast, and all of those model organisms were sequenced using a clone-by-clone approach, in which chunks of the genome were cloned in bacteria and propagated, and then methods could be used to select individual clones that span the entire genome and sequence these in a piecemeal fashion. And what I'm showing on the slide, oh my goodness, I have many, many options. What I'm showing here is one of these beautiful back clone fingerprinting gels. And so one could generate many hundreds of thousands of these fingerprints. Each row is a restriction digest of a bacterial clone. And by having a unique fingerprint for each clones, the fingerprints could be compared to each other and overlaps could be constructed. And then what you would do is you would pick a tiling path, a minimally overlapping tiling path across this region. You'd sequence it and you would say, I have representation of the region. And this was done using, at least initially, manually loaded slab gels. We won't go into that. And then later on, capillary gels, which were the revolutionary technology that Craig Venter gained access to first in the United States, which made a lot of money for ABI eventually because everybody needed those things to compete with Craig. So the reference genome era was useful, in my view, because it generated reference genomes. And it turns out that in the alignment era, this was the key resource. So methods, a key output, actually, of the Human Genome Project, besides the sequences of these various organisms, including that of human, was the realization that technology could be made where you could, in a massively parallel way, generate short little tiny sequence reads. They in of themselves could not be reassembled into the complete genome or a facsimile of it, but they could be compared and aligned mostly uniquely, except for repetitive sequences, back to the reference genomes. And so this really was the start of an appreciation for the fact that we now had a tool that we could sequence genomes and resequence genomes again and again and again and begin to understand the landscape of variation, not only in human species, but in species that exist in nature. And we could align variation that we detected using this approach to health conditions, to phenotypes of plants and animals, and so on and so forth. So that reference genome era spawned the alignment era, but we've sort of come full circle. Now we're in the long read era, where it's become evident that through certain technologies that have been available for some time, we can generate reads spanning tens and even hundreds of kilobases in length, and these then can be reassembled and read back into complete sequences. And the reason we need to do this is because the alignment era showed us that we could only capture some of the variation, and often not the most medically useful variation. We needed better tools. The long read era has also allowed us to think about other types of information encoded in genomes, not just the base compositions, not just variation at the level of the single nucleotides or larger variation, structural variants we call them, but also DNA covalent modifications such as methylation. And so we can read out methylation, and what's shown here in a almost undecipherable glyph is some blue stuff right there and some red stuff. This is one haplotype, so this is a human sample. We can build out an entire haplotype, so the allele that was inherited from mom, for example. We can build out the allele that was inherited from dad, and we can see differences in the covalent modifications. And if you focus in on this particular region here, this little tiny blue sliver, and you blow it up, the two haplotypes can differ rather markedly in very important areas that control the expression of cancer genes. So that's the era we're in now. We're still using short reads, and we're just starting to use long reads. But it's in this context that we now have the opportunity, the technology, but do we have the will to start to apply this kind of technology systematically to uncover the variation that underpins most of life and is so important to understand in various clinical applications going forward? That was my 25-year summary. Thank you. So variation becomes the thing that we want to understand, I believe, and cancer is a good model for this, because it's an incredibly heterogeneous disease. There's a heterogeneity of phenotypes And these heterogeneous phenotypes have as their underpinnings variation in the genome, the transcriptome, the proteome, the epigenome, and fundamentally we need to uncover those pieces. So this is a very famous picture taken from Hallmarks of Cancer papers published by Hanahan and Weinberg. And what it shows is that cancer tissue is a heterogeneous mass. There's vasculature, there are normal cells, there are tumor cells. The tumor cells are not necessarily all of the same genotype. They have different genetic makeups. It's a right proper mess for the most part. And because it is a right proper mess, and because it varies over time and in space, even within an individual's cancer, we have to come to the realization that for the most part in cancer, every time a selective agent is applied to this, a chemotherapy, a radiation therapy, every time we apply a selective agent and a cancer cell escapes, we have to be cognizant of the fact that genomically it may be different and that will seed a new cancer. And so this sets the stage for what is really, as it's been called, the emperor of all maladies. So targeting this heterogeneous gemisch is a challenge. And so the first step has been to identify features that drive malignancy, and that's that colorful series of glyphs on the outer ring. These are all the phenotypes that cancer cells have in order to be cancer cells, and targeting these are a heterogeneity of different drug options, which are constantly in development and evolving. At least that's what we hope. So we have this heterogeneous mess, which is the tissue. We have a heterogeneity of phenotypes, a heterogeneity of treatments, and we need to understand the underlying genomic heterogeneity. Not that it will necessarily be the complete things that we need to understand, I mean, but it is a tangible and feasible thing to start to think about. I have a conceptual slide next, which presents a medical oncologist's view of the problem. So imagine a population of patients. Those are the patients. And in this population of patients, you could sequence their DNA. And when you sequence this DNA, you may be able to find common properties among selected groups of them. So the red people have a commonality, and the green and the yellow and so forth. And then the idea would be that you could use sequencing as a screen to stratify patients into bins, and that this might enrich for the populations that will respond to particular agents. So that's a nice concept. And I think this is a useful slide to think about. The problem basically is, as we illustrated here, the scale of the thing, obtaining the material to do the sequencing. This is only possible through the active involvement of the clinic and clinicians in it. The idea that you could align these things to drugs depends on the drugs that are available for the condition. And if you constrain your thinking to the drugs that are available for a particular indication, I think you're missing the boat here. And the POG program, which I eventually will get on to talk about if I hurry up, will show a little bit about how off-label treatments become a very important thing to think about, particularly in the cancer context. So the clinic has embraced heterogeneity in cancer to a certain extent. And we can see that please don't read this, it's not meant to be read. But what it is meant to show is that the clinic has embraced the idea of heterogeneity and that genetic underpinnings matter. And so we have these things called panels, which are limited interrogations of a few genes or variants within them. And this can be done in the context of somatic cancer. So the Onco panel tries to find cancer mutations that can be aligned to approved drugs for the indication. The focus panel is a similar sort of thing. And then there's the hereditary cancer panel, which considers a much larger list. And many of the things on this list are known to be significant in the context of cancer. So these are moderate to high penetrance alleles, if you like, that confer a measurable or even substantial degree of risk. And you want to know about these things because these are the things that run in families and can predispose individuals to malignancy. So this has been recognized. It also illustrates a few of the challenges here. And I think one of the bigger challenges is the discovery space for cancers. So we can look at a series of cancer patients sequenced as part of POG. We call this the POG 570 cohort because there's 570 patients in it. These represent a highly sanitized, very carefully analyzed group of patients. And what we know from the 570 patients, and this is the third line down under somatic substitutions, is there's an average of about 13,000 somatic mutations per patient. So these are mutations that occur in the DNA, but don't occur in the normal, so somatic mutations. So we have this very rich and actually quite fearsome array of somatic mutations. And we have to be able to understand within that cohort of somatic mutations, which are the ones that we might take advantage of somehow in the clinic. So that's the challenge. We have a few genes on contemporary panels. We have many, many mutations in many more genes. How do we reconcile our path going forward? Clearly, we're undersampling somatic mutation space, and clearly not all of these somatic mutations have the potential for therapeutic benefit. But there they are. And it's based on the phenotypic properties that these mutations encode that allow cancer cells to be cancer cells, if you think back to the original picture. So that was the problem that we recognized very early on in the genesis of the POG program. And so the question became, well, can potentially actionable cancer vulnerabilities be routinely identified using what was then the most comprehensive approach that we could think about that was actually feasible and scalable? So genomics is feasible, it is scalable, and it can reveal these things. We wanted to do the whole genome, not panels, because that was the only opportunity that we could think of to cover the entire space within a patient. There were many, many criticisms, continue to be, about all of this kind of thing, but I'm convinced, and I think others are too, that you have to capture the whole thing as best you can, an approximation of it, if you want to capture all the variation and have the potential to leverage it. So we came up with the personalized oncogenomics project, and this started to get rolling at about 2012. And it was very important that we have a wide cross-section of leaders with complementary expertise as part of the POG program. And so this goes back to the concept of leadership. The POG program could not have started, at least not the one that we envision today, if it was a couple of guys having coffee thinking about the next cool genomics project. That was impossible. We needed people in the hereditary cancer space. Intan is there. We needed people in the medical oncology space. My co-lead is a medical oncologist, and we have others. We needed pathology representation. We needed health economics. And of course, because this was a massive data generation activity, we were changing samples into data. We needed to have bioinformatics and cancer informatics more specifically. And I'm pleased to see Steve here today. Good morning, Steve. And excellent project management. We shouldn't diminish the role that has played, because without Jessica, none of this would be possible. And so together, this leadership team compiled a group of interested parties who came together to drive the project forward. And so there's a bunch of medical oncologists, a huge number of folks from Canada's Michael Smith Genome Sciences Centre that have participated in and driven this thing from the very beginning. And the point is not to go through this in any great detail, other to show that the program today is an institutional program, and continues on, and albeit at low level, as a very important way for us to probe variants and the therapeutic vulnerabilities that they confer. So the program is organized thus, and at this point I've put up a bunch of symbols because I want to acknowledge the support that POG has had, and these are but a few of the more recent ones. And so BC Cancer Foundation and BC Cancer have been supporting POG since its outset. Genome British Columbia came on relatively early in this process to support POG, and it's been absolutely critical as a way to drive the genomics agenda forward, so that's wonderful. Most recently, the Terry Fox Marathon of Hope has recognized that POG has much to contribute to the network that it's built and is trying to continue, as well as UBC. And of course, the Canada Foundation for Innovation has supplied the infrastructure. So the complexities implicit in the organizations involved in supporting it and the complexity of the disease itself have appeared fearsome at times, but but this is what we see it takes in the country is this broad coalition of support at the scales that we're doing anyway. So the medical oncologist will identify a patient and enroll them onto study. A biopsy is obtained when it can be safely. Most recently, we've been reflexing onto formalin-fixed and paraffin-embedded tissues that are commonly collected for pathology purposes. Those actually are okay, but largely awful in terms of their genomic properties. We use them anyway, and we have become aggressive about biochemistry and bioinformatic means of trying to filter the formalin-fixed, paraffin-embedded artifacts. Remembering that these tissue blocks, as they're called, can sit in a shoebox for 25 years under somebody's desk before we might find them. And that's not really how you want to preserve nucleic acids for the kind of analysis that we're doing. It's not because they can't perform well. It's often because of how they are stored. Anyway, biopsies are best, but not always possible. Whole genome and transcriptome information is made available, and the patients have all consented to data sharing, publication of the information, and access of information through controlled access sites. So that's important, actually, because that's the way that we get the word out, is by talking about our experiences here, sharing the information with other jurisdictions, most of which actually are not in the United States. Reports have been designed and are generated, so designed by clinicians and generated for clinicians. And there's two kinds of report. There's a targeted report, which we aim to have out the back end of the Genome Center within about 2 weeks of receipt of the sample, and that needs to be whittled down further, but that's pretty good. I mean, if we can hold that line or improve it by a day or two, that's pretty good. That targeted gene report is essentially a glorified panel report where high-value, obvious targets are identified early. It's not the full genome report that comes out after a longer period of time, very much a bespoke analysis of the tumor properties. But the existence of the report facilitates an interaction with the molecular tumor board. This is now running, I believe, twice a week. And only a few patients are dealt with in that particular context because it's a time-intensive thing. And what we're trying to do is take a very complicated genomic profile aligned to therapies as best we can and communicate that to a user audience, in this case our oncology community, 5 to 10 minutes. And so that data reduction challenge is a big one. And it speaks to the difficulty in scalability as POG is implemented now. It's not scalable, and we have to think about that. Okay, the germline is an important piece of this. And so because we're sequencing the tumor DNA and we're sequencing the so-called normal DNA, and we're comparing the two things and reading out all the variants that occur in the tumor, but not all the variants that are occurring in the normal. And remember, there's something like, I don't know, 3 to 10 million variants in your normal DNA. The copy of the genome you got from mom and the copy of the genome you got from dad are very, very, very different things. So you are a heterogeneous mix of variants. Basically, you're a mess. But we have ignored that largely within POG, at least not dealt with it well. And so the germline becomes, I think, a very important early target in all of this. So we know from POG, and I don't know if Intan I don't see here, that's Kasmintan Schroeder, who's on the author list here someplace. Intan tells me that between 10 and 15% of patients we see in POG identify a previously unknown hereditary cancer family. So those are people that are carrying around a moderate to high penetrance cancer allele, which elevates their risk, and they don't know about it. And so when we sequence the normal, we can find those things. That's in the cancer context. This is a very, very beautiful study that we heard about when Dr. Yvonne Bombard came to visit us just a few weeks ago. And what Yvonne has done is take exome sequences from a cancer population and asked, in those exome sequences, are there other secondary findings that could be medically actionable? And I think this is really, really important. And the answer is yes. And so it turns out that of all the participants in the study that elected to receive information on these variants, all of them had at least one. And this is not in the cancer context, okay? This is outside the cancer context, which was the purpose of generating the sequences in the first place. Most of the variants detected related to pharmacogenomics, drugs, if you like, and the ability of the individual to receive them or not. What was, I think, especially interesting is that knowledge of these variants through the network that Yvonne established, and about 28% of them, they actually changed management. 28% of secondary findings were actually acted on by a medical professional in some context or another. So when we think about, precision cancer genomic medicine, and we're often thinking about the tumor, I think that's too narrow. I think we need to think about the germline as a very early win in all of this. And many of the projects being funded in CPHI, well, all of them, I think, are focused on issues of the germline. This is an amazing opportunity for us to advertise our findings, relate them to as diverse a number of healthcare contexts as we can, and bring additional benefit for dollars spent, and nucleate the genomics and healthcare communities. We have all these silos in BC about, oh, this is cancer, or this is cardiac, or what have you. The genome doesn't care about any of that. And the comorbidities that modern populations experience, they're all read, or at least a substantial portion of them are read in the genome, and 28% of them can be acted on, hopefully to the benefit of the patient. This to me is the thing we must do, and we're not doing very well. It's a nucleation opportunity, a community building opportunity. Okay, so back to POG now. And this is an example of a lesson that somatic cancer genetics learned from the germline. And so this is a patient with a very aggressive head and neck cancer, and the genome was sequenced, and this fancy little diagram here, I'm not going to go through because I barely understand it. I've been staring at them for almost 15 years. But the point is that red means hot or up, and blue means cold or off or down. You can think of it as loss of function mutations in blue, red is gain of function, red is increases in expression, all of that sort of stuff. And so there's many targets on this slide. This is predominantly oriented towards those targets. The big red arrow is pointing to a gene called DPYD. And there's this big crosshatch, which indicates that there's some sort of fusion that is inactivating DPYD, and it's homozygous. That's what the glyphs mean. So DPYD was interesting because we know that it's involved in 5-FU metabolism. And we know in the germline, if you have a DPYD mutation and you get 5-FU, you can get very, very ill. In fact, you can die. And so screening for DPYD in the germline becomes very important. And not all jurisdictions in the country actually do this, but all of them should, obviously. There was a very high-profile case of a physician who passed away because they received 5-FU, did not know about this. All right. So this patient's germline was fine, didn't have a DPYD mutation, but their tumor did. And this led to a hypothesis. And the hypothesis was, well, okay, if DPYD mutation is toxic to human cells, the normal cells don't have it, the cancer cells do, maybe the patient ought to receive 5-FU. They did, and they had actually an incredibly significant response where the tumor had just melted away, whereas before it had been growing under state-of-the-art, very fancy and expensive immunotherapy. Okay, so POG has done a lot of stuff. I don't think we need to go through all of this, but I will go back to this concept of leadership and nucleation and bringing communities together. And that's 83% of cases presented at POG Tumor Board have a clinically actionable finding. So the hit rate is very high. These are clinically actionable as adjudicated by the oncologist managing the patient, not by Dr. Jones or myself imagining that this might be a good thing. This is the oncologist talking. Despite that fact, relatively few are acted on. And that's due to two things mostly. One is the stage the cancer patient is at when they become available to POG. It's usually very late in their journey. And so we have to do better there. We have to get to people earlier, people at high risk. I agree that the scalability, until we can do very, very large numbers of people, remembering that there's going to be 35,000 new cases of cancer in BC this year or next, we can't do them all right now. Maybe one day, maybe soon. So we have to segregate somehow. So high-risk cancers make a lot of sense. Getting them early in their disease trajectory makes a lot of sense. Acting on the information that we find makes a lot of sense. And being able to show survival benefits in that population is the next most important step. 132 medical oncologists or fellows have enrolled patients into POG in the last 3 years. So that's a huge number. That is the vast majority of oncology, medical oncology practitioners or fellows in the province. And that is the most important thing, because in doing this whole exercise over the last 12 to 15 years, I think what we've done, more than any other single thing, is we have trained a community of oncologists to embrace data at scale. And cancer is a data problem. It really is. 13,000 somatic variants in everybody's malignancy, 3 to 10 million variants in the underlying germline. It's a data problem. So if oncologists or practitioners in any field embrace the data challenge, if scientists can figure out a way to make it embraceable, then I think we can go forward in a very meaningful way, and we should. Okay, the POG platform has become very popular. Many projects use it and relabel it according to their preferences. And so there's pediatric patients represented here. POG platforms are used for something called Profile, which is pediatric. There's a PEDS POG arm, so pediatric cancer patients in that arm too. Patients are stratified to trials using this now. So patients have access to new drugs that are being tested because they have variants that align to the indication. All right, and then we go on to the Marathon of Hope. So this is quite a story, and it really gets, I think, to the heart of what it takes to build a community. So the whole concept behind the Marathon of Hope goes back to a fellow named Victor Ling, who was Vice President of Research at BC Cancer for many years, who then founded the Terry Fox Research Institute. And the research program that eventually became a sort of a hallmark was the Cancer Centers Network. These were discussions that started happening in the Harper government, for those of you that are old enough to remember that. And so Vic persisted when things didn't work out with the Harper government, went into the next administration and then the next administration after that. And finally what happened was $150 million was forthcoming from the federal government to be matched on a one-to-one basis by jurisdictions, partners, if you will, across the country. And so today, the picture illustrates how the project is organized. So there's participants in BC, there's the prairies that are considered a region, Ontario, Quebec, and the Atlantic. And so far there's 42 members in this, and these are members that contribute in some way, and the lists are still evolving. So what does it want to do? Promote collaboration, data sharing, knowledge and technology insights. It needed a project, it needed something to do, not just a collection of people that said, yeah, we believe in this. So the question then became, well, if we're going to share data, what data are we going to share? What data could we generate in the Atlantic provinces that could be analyzable and understood by people on the West Coast? And so genomics was the answer to this. And the other reason genomics was the answer to this was because already in clinical practice were well understood markers that would lead one to manage therapy in some way or another. So a proteomic profile that contained things that weren't relevant to medical oncologists would be unusable. You needed mutations. You needed variants, and ultimately it turns out you need the transcriptome too. That's not all you need, but you need those things. And so it became whole genome and transcriptomes like POG, and was heavily motivated by POG. And the idea then became, well, we need a number, so let's do 15,000, not because it's a rational number, but because we need a number. And we needed to show that we had a target and we could deliver something to the target. So that became a reality about 5 years ago, and the project started. So this, This is where we are today. So we said we would generate 15,000 cases. We have less than a year to finish it off. We're slightly more than halfway there. The scalability problem has been a challenge, but we do think that we might be able to achieve this. And so you can see there's 5 consortiums, 42 institutions, 120 contributing cohorts. So a lot of the work that's been done historically has been done using retrospectively collected samples. So these are samples that were in somebody's freezer that were fit for purpose, not necessarily to align these profiles to medicines. That wasn't the objective in the first phase. The objective in the first phase was to come together to share data, to understand how we could collaborate, to get all the legalese and crazy things out of the way, remembering that each hospital is an entity unto itself and there's a zillion of them across the country, never mind each province. And so battling this is ongoing. By the way, it's not completely solved, but it is largely addressed, actually. And there are data sharing systems in place. And I'm happy to report that BC is leading the way in sharing data of this sort. And POG has been doing this, of course, throughout, providing a path forward so that it can be understood that eventually there will come a time when we will want to analyze these profiles and align them to medicines that people can get. What else to say? Probably not much. This is too complicated to dwell on, other than to say that we're generating germline sequences, we're generating tumor sequences. All of this is collated with clinical data, which has to be extracted, sometimes in a very manual way, from hospital databases, sometimes from paper charts. So a huge data collection initiative, and then all the problems with how to release the data. And the business of how to release it is really illustrated down here. So what's been decided on is that there needs to be a federated method of data release so that jurisdictions like BC or Montreal or what have you will be able to release the data that they feel comfortable releasing, that the various lawyers and so on and so forth that are involved in all of this are comfortable with them releasing. So each jurisdiction makes up its mind about what data that it can share. Data sharing will be, in the first instance, with members of the network and then beyond. So provision for all of that is taken into account. So that's what it's looked like. And if you want to read more about it, we were very lucky to publish a perspectives piece just very recently in Cancer Cell. There's a huge amount of other information here. There's something like 15 working groups, each with 5 or more members who are talking about standards and data alignments and guidelines made up from people from all over the country. Huge numbers. There's a patient working group. There's a focus on how do we find and include underserved and underrepresented populations. All of that. I've dealt with almost none of it, but if you want to know more and you want pointers to sections on the website, including some very informative guidelines around various things, please do have a look, and hopefully you'll find what you're looking for. Okay, so POG has done a bunch of things, and I think one of the most important things is it's provided a way for us to think about the value of new technologies going forward. And now we have to go back to the essence of what is cancer. So the new technologies that are the most use may or may not be suitable across all indications. We don't know. But what we do know is that cancer is a disease fundamentally of single cells, which then expand. It's highly dynamic, and the cells can all be very different. And we think the seeds of treatment resistance lie within the variation, the genomic variation that creates phenotypic variation that can then be selected on. And as soon as a cell escapes that selection, it's treatment resistant. And if that can find its way to a location where it can recolonize and establish a new cancer, it is exactly that. It is a new cancer. So the cancer solution in terms of technologies is not necessarily the same solution that we'll see in cardiology or nephrology or what have you. So we think about this in the context of the variants that we want to detect. So long reads become important. We think about this in the context of single cells. And then a topic that is very dear to my heart that was created by Dr. Yuka Takamon, who's in the audience, so you can ask Yuka about this, is synthetic lethal analysis. And that I'm not going to touch on it today due to time constraints. But here, what we're trying to say is we don't care about the 13,000, the actual functions of the 13,000 mutations in the cancer genome so much. We just care whether they make a cancer cell vulnerable or not. So we're seeking to capitalize not only on the mutations that are specifically aligned to approved drugs, we're trying to find every Achilles heel in the cancer that we possibly can find. And I think Yuka has created a very nice way for us to do that in a data-intensive way. Talk to Yuka. So the problem, right now we're doing bulk analyses. So we grind everything up and we get our smoothie. But hidden in the smoothie someplace are a half dozen blackberries, and we'd like to find the blackberries because it's the blackberries that are the bad guys, as everybody that hates blackberries knows. We want to be able to ferret them out and understand what their properties are. And if we can understand what their properties are, then we hope we can defeat them. And so the single cell approach basically takes a tumor sample and busts it up into its constituent cells, where each cell is analyzed not at a comprehensive level, not yet, but at a very, I guess, superficial level almost. But enough information is generated to be able to differentiate cells from each other. And then there's the business of spatial analysis, and that's the flan. Is that a flan? I don't know. Anyway, the flan-looking thing on the end, where not only do you understand information about each cell, but you understand the spatial relationships of cells with each other. And this becomes very important, we think, because in cancers, if you have a nice immune cell, which is actively primed sitting next to a nasty cancer cell, and you can cause the cancer cell to be recognized by the immune cell then has the opportunity to act on the cancer cell. So the proximity of immune cells to cancer cells becomes important, and spatial profiling is probably one of the better ways to realize this arrangement of cells to each other. So the smoothie approach is still important because it's the most comprehensive data, but you can layer on top the fruit salad and the flan approaches on top of the genome. Here's an example. So this is a patient that came into the POG program. Their malignancy is not entirely relevant. It was a thymoma. But this particular patient had a tumor content of 23%, and this is the face page of the report. We know where the biopsy came from. We look for things like microbial species. There's an immune cell score that we provide these things. But the upshot here is very low tumor content, 23%. So just imagine you're doing a gene expression profiling experiment, and only 20% of the cells in your gemish are the cells that you want to recover. When you make the smoothie, you can't find them, at least not very well. So that's a problem in this particular case and in many others, because there's gene expression that could represent important targets for therapy. And now what you don't know is what cells those genes are expressed in. Are they expressed in the tumor cells? And if they are expressed in the tumor cells, then perhaps they become legitimate targets, things that we could target through an approach using drugs. But if they're in the normal cells, we don't want to do that for obvious reasons. So what cells are these targets in? And that's where the single cell approach can help us out. And that's what we show here. This diagram is the much unloved UMAP projection, and there's all kinds of reasons why we hate UMAPs. And those in the field know exactly what I'm talking about. But I don't think anything better exists for the purposes of this illustration. So we'll call these UMAP projections as what we're going to talk about. Each little dot on this projection is a single cell that has been profiled using two technologies. One is called ATAC-seq. Doesn't matter what its name really is. It looks at the chromatin and its activation state. And the other is single cell RNA-seq. And so what we've done is we've said, okay, where are the tumor cells in this gemish? Oh, and the relationships of the cells to each other don't represent their spatial relationship in situ. They just represent the convergence of similar properties. So cells that are in the same grouping are more similar to each other than cells that are more distantly arrayed. Okay, and so the ATAC-seq samples little regions of open chromatin. And because we had the smoothie, we had a bunch of mutations, we could look for the presence of somatic mutations in the ATAC-seq reads, which are very sparse, very, very sparse. They barely cover any of the genome at all. And we could find them. And when we found them, we colored them red. So those are mutation-bearing cells. And because they are somatic mutation-bearing cells, they're thought to be malignant cells. And you can see that the majority of these cells are clustered here. And so we say, well, that goblet-shaped thing is an enrichment for the tumor cells. And then we can say, well, what are the cell types that we see? And for that, we use the RNA, and so we can map the various cell types. We call these the tumor cells, but you can see there's T cells, neutrophils, fibroblasts, all that sort of thing. And what's kind of amazing, actually, was when we looked at the proportion of cells that we had classified as tumor, it was 22%. Remember, from the bulk analysis, it was 23%. So what's so amazing to me about all of that is that we managed to get the tumor content estimates right, even though only 23% of the cells were in fact tumors. So that's kind of amazing that the bioinformatics is that good. But now we know exactly where the tumor cells are. So once we have this, we can ask ourselves, okay, well, where are the targets expressed? Audience participation time. So let's look at PD-L1. This is a very famous target for immune checkpoint inhibition. Okay, Stuart, you've been paying attention. Tumor cells or not?
Sure not.
Yes, that's right. Good target or not?
Yeah.
I like this guy. He's great. Thank you, Stuart. That's exactly right. So that resolution then led to, as well as IRS4 expression, led to the realization that these actually could be rationalized as being legitimate targets using therapies that were on the shelf. And then CDK6, you can see, is not preferentially enriched in the tumor cell population. So in this way, we can imagine starting to layer in a new kind of technology to give us additional information that we can't get out of the bulk. And even in clinically relevant low tumor content samples, we have a way to think about accessing a drug aligned to an indication, whereas before we couldn't with confidence do that thing. Okay. So, a few things in closing, really. The Marathon of Hope project exists because there has been a national convergence on trying to solve a problem. It's confronting some very, very big challenges in the national context. How do we come together? How do we share data? You know, how do we set common standards across this huge country with all these different healthcare organizations? How do we motivate leadership to continue to participate? All of this are challenges. And fundamentally, what makes it all work is funding. And so funding becomes critically important. Even funding as diverse as what we see in the Marathon of Hope, where BC Cancer Foundation, Genome BC locally are the major contributors. We get money from the federal government through Terry Fox. Every single hospital participating in this and every single jurisdiction has its own funding constraints. So that's part of the problem we have to confront. It's a practical one, but we need the money. We all need the money. And if we don't have sustained investment in genomics at scale, we will do very nice proof of concept experiments in each of our particular diseases of interest, and we will make contributions, and we will not realize the value of those contributions unless we can translate them somehow into the broader context. So this business of translation cannot be done by a couple of scientists in a lab. It really does take a community. It really does take genomics leaders who believe in application leaders who believe in the genomics, and in this case, our application is health writ large. So that's a lesson, sustained investment. And believe me, with all the nonsense that's going on elsewhere in the world, this is not a time for British Columbia or Canada to relax. It's quite the opposite. There's an opportunity for leadership that hasn't existed in North America for a very long time. Let's not waste it. In terms of the Marathon of Hope, we're now moving on to phase 2, or at least we hope we are. And in phase 2, the objective is much more like POG. It's to generate a profile and return a result. Return a result to whom is an open question. I would argue that if it needs to go back to medical oncology and pathology and healthcare professionals if we are to act on it, because that's the established route, that's the access to drugs. So minimally, that has to happen. Achieving that nationally is a huge challenge. People are buying into this so far, notionally anyway. If this does come to pass, it'll probably be on a smaller number of cases because we'll have to show that we can do this. But just imagine this. Imagine that Lillian Sue, in her office in Toronto, has just seen a patient. Imagine that Lillian has a profile for that patient, doesn't really know how to interpret it, goes into the central database, finds a patient with a variant, maybe a similar disease that was acted on in BC. And that information can be obtained by Lillian sitting at her desk in 5 minutes. What would that do? What would that do? I think it would help. And I think it would be profound. So that's the whole thing around phase 2 is the business of creating that opportunity. And it's not to bring it directly into healthcare, because we can't do that. But it's to do the research that would present compelling evidence that healthcare would need, and to make our story known so that we can get advocacy, because advocacy is incredibly important, so that we can bring this vision to reality. There are many people to thank, many organizations to thank. I can't possibly do it all. I'd like to start with the funders of POG. There they are, for the most part. There are others. Infrastructure support has come about through various means and ways. And many organizations in the province are starting to participate either in POG directly or in the Marathon of Hope. None of this would be possible without an organization that does large-scale, high-throughput genomic science. So the Michael Smith Genome Science Center is fundamentally critical to carrying this phase forward, as are the genome centers in Montreal and in Toronto and other places. Jurisdictions have to have these things. They have to. We can't do the research that we need to do in order to motivate the healthcare systems to make this happen without this kind of infrastructure. It's critically important. And of course, along my own path in genomics in many, many years, we haven't talked about forestry, we haven't talked about fungi, we haven't talked about fish, we haven't talked about any of those things. But there has been a community of people that have funded genomics in BC, both within and outside of the country. And I hope I'm not speaking out of place when I express thanks to all of those people for all their support of genomics over the years. And then there's my own lab, who I love dearly, and several of whom are contributing to POG. This is the MATCH lab. We style ourselves MATCH after multi-omic analysis of treatment, resistance, and cancer heterogeneity, and that's why we call it MATCH, because it's way easier to say. And then, most importantly, the patients that enroll in the program, that believe in the program, that advocate for the program, that want to see the program continue and have the force of will to drive it forward in corridors of government that would make scientists uncomfortable. That's why we're here, and that's why we exist. So thank you all very much.
I'm going to try it works. Excellent. I'm going to try the microphone. So I'll stay here. Thank you very much, Marco. That was quite incredible. So we have the, we are it's a little bit behind schedule, but still I think that you mentioned some very important things is worthwhile just reflecting for a minute. While we do that, you have the polls result up there. Okay. This is very interesting. Now, I have my own opinions on these results, but this is very interesting. Marco, I don't know if you see them, right?
Yeah.
Yeah, great. So I think I'm going to ask you a question which sort of reflects a little bit on these results. I think you did a fantastic job to illustrate why all of the people in this room and online believe, or why they believe that implementing whole genome and transcriptome sequencing in routine oncology is necessary. You also showed that very well, I think, that the path is extremely complex and it doesn't happen overnight. And I think we increasingly get asked, how do we accelerate the progress towards this scaled implementation? Which is a really important question, and it's a good question. But I think there is a little bit of a gap between the people in this room, the people like yourself who are doing all of this, are demonstrating, are in the middle of this path, and obviously the people that would like to see this scaled up for the benefit of patients and implemented in routine cases. And so I wanted to ask you on practical terms. You mentioned already two, actually. You mentioned leadership at the beginning of your talk, and you mentioned another one at the end of the talk, which is sustained funding. I think those two things are super important. But perhaps you want to expand a little bit on what is your view in which we accelerate this, because this is very complex, and it takes a village, a community, as you said. And it is necessary. So
over to you. And you've seen some of the polls results as well. I mean, all of those things up there are in fact necessary.
So there's a list of things that people were meant to select. And when I look through this list of things, I see all of them represented in what we've been trying to do, all of them, not this one over this one, but all of them present in all of the thinking and all of the hard work that over the past, 12 years, we've had to confront both locally and also nationally in the Terry Fox Research Institute. So it's this list, yes, everything is important, of course it is. And there's room for all of it. There has to be. There's the research, there's the implementation and scaling, and then there's the application. And each one of those kinds of things has a separate list of requirements. And so the way that we've thought about this in the Marathon of Hope context is, we have a vision. The vision has been embraced by a large number of people within the network. And actually, if you don't believe in the vision, then you have to ask yourself, why are you part of the network? We don't even know whether the network will continue, by the way, except, spiritually or in our hearts. But we intend that it will. So how do you drive forward, recognizing the diversity of the research that needs to be supported? How do you drive forward the agenda? I think, and it's unfortunate, but we live in times where it's very difficult to separate politics from funding. And we also, I think, need to establish some venue where politicians can listen to researchers, understand in some way what's being said, and be shown the value. And this is not something we systematically do, or at least I don't think we do. We have these bodies, these august bodies, Royal Society of Canada, this, that, or the next thing. Do we have here in BC, do we convene as a group of Royal Society of Canada members to talk about what the biggest challenges are in health and genomics? And if we were to do that, how would we communicate a consensus view from that group back to government? What's the communication channels? How do we talk to government? How do we say this is a good thing? And this is maybe not a good thing. How do we do that? Do we have a venue for that sort of discussion? And are those of us that work in healthcare environments actually comfortable in participating? Because, very often we're discouraged from talking to government. I don't buy any of that. That's death. The scientists are the idea factory. They have to be the idea factory. The researchers have to research. They have to. That's where the product is. And the best solutions have to come from the people doing the work. The implementation then, and the scale of the implementation, that has to be communicated in some way to funding bodies. We've been to Ottawa. We talked to MPs. We talked to people higher than MPs. We talk to advocates. We have to be out there. We have to be talking. We are talking in Ottawa. We've done quite a bit of that. Are we talking in BC? Now, BC does a lot for research. I'm not being critical here. I'm just saying that if we want to motivate this, there has to be a more advanced understanding of what it is that we're trying to do, why it's important to try to do it. But most of all, we have to be trusted when we say something. If we say a thing is a thing and nobody believes us, what's the point in saying it? There isn't any. I think this business of recognition of the credibility of the scientific community may be an issue. How do we build it? Anyway, the answer is not easy to provide, and it's certainly not easy to do. But it doesn't help if we're not all part of the same house at some point in our discussion. We have to be able to sit down and say, this is important, this isn't important. What is the correct framework for doing that? It has to have an outlet. Who do we talk to in government? Do we all do this? Do we just all kind of like camp out at our MLAs or our MPs' offices? Is that how we do it? Do we organize? How do we do it? Or do we just wait for the next funding call to come along and say, oh yeah, I could kind of take advantage of that thing and I could do a project. We don't have programmatic funding in this country for the most part. We have project-based funding, and it's meant to go away. And then here I'm talking about a program that has gone on, 15 years. How do you get that done? It takes 15 years to do anything. So how do we do that? Or are we going to just do 4-year projects and we're going to say we've solved the problem, which is absolute nonsense when it comes to this kind of implementation. If we're talking fundamental research, sure, we can do projects. But we have to think in terms of programs, how to advance them. I'm just being too windy now, so I better shut up.
Now, this is great, Marco. It's exactly what I wanted you to say.
I didn't know that.
You fell right
Honest.
into the trap that I wanted you in. So this is great, and I hope we can all in this room at least continue this conversation with Marco during the breaks. So thank you very much. So maybe an applause again, and thank you. Thank you to our audience as well, here and online. We're going to have a short break. AG, is it still going to be a 10 minutes short break? Yes. Okay. So we'll continue our first panel session of the day, which is going to be focused on shaping maternal and child health right after the break. So for those online, all you have to do is return to the event hub and click on our next session to rejoin in 10 minutes. So that would be 9:40. Have a good break. Thank you.